Two questions have defined the Indian subcontinent's history: the origin of its first city builders and the timeline for Indo-European languages. Until recently, resolving this required navigating a conflict between linguistic reconstruction, fragmented pottery shards, and ambiguous textual references. This reliance on indirect evidence ended at Rakhigarhi. By targeting the dense petrous bone of an individual designated I6113, researchers successfully sequenced the first complete genome from the mature Harappan period. This single Harappan individual provides the anchor for a massive comparative dataset of 523 ancient genomes from across Central and South Asia. These sequences provide a biological baseline for the Harappan population, replacing stylistic archaeological categories with specific genetic data points. Geneticists interpret these data points through probabilistic models that measure distal ancestry, the deep lineages that branched off thousands of years before the Bronze Age began. Bypassing generalized historical summaries requires an analysis of the specific component-level architecture found in these ancient samples. Because human history involves continuous mixing, geneticists use a statistical model that simplifies ancient Eurasian complexity into 7 deeply divergent distal sources, each assigned a specific color. Tracking these 7 specific components across time reveals exactly how the Indus Valley genome was constructed and the specific moment its composition shifted. The woman from Rakhigarhi shares a nearly identical profile with 11 genetic outliers found at trade hubs in eastern Iran and Turkmenistan, establishing a consistent Harappan signature across the region. This signature is a mixture of 2 lineages: the ancient Ancestral South Indians, represented in yellow, and a blue, Iranian-related hunter-gatherer component. The Harappan baseline contains 0% Anatolian farmer ancestry and 0% Western steppe herder ancestry. Understanding the blue component requires tracing its lineage back to the terminal Pleistocene. Statistical distance metrics prove this blue Harappan lineage diverged from the ancestors of Zagros mountain farmers more than 12,000 years ago. This deep time split occurred long before the start of agriculture in the Near East, indicating that the transition to farming in the Indus Valley was not driven by a mass migration of Neolithic farmers from the Fertile Crescent. The absence of Western steppe DNA confirms that the mature Harappan urban phase predates the arrival of Indo-European speakers in the subcontinent. Tracking the steppe ancestry that is missing from the Indus Valley requires shifting focus northward to the Bronze Age pastoralist roots. Independent researchers have applied intense scrutiny to how the 7-source model maps these migrations across the Central Asian data. Much of this friction involves the ancient samples from the Bactria Margiana Archaeological Complex, or BMAC. These BMAC samples show significant levels of Anatolian farmer ancestry, identified in the model by the color orange. The current genetic consensus suggests that steppe pastoralists bypass these established BMAC populations to integrate further south into India. However, examining the raw color codes from the gateway into South Asia reveals a potential discrepancy. By 1200 BCE, ancient individuals in northern Pakistan's Swat Valley provided the first genetic look at the population entering the subcontinent. In the simplified 7-color model, these early Swat samples register a combination of blue, orange, green, and yellow. This creates a specific point of debate. If the steppe migration bypassed the BMAC, why are the steppe-specific colors of teal and red initially missing from Swat, while the BMAC's Anatolian orange is present? Resolving this friction requires moving beyond the surface-level color codes of simple bar charts. While color charts provide a visual summary, they can mask the complexity of how ancient populations actually mixed. Geneticists use a statistical tool called QP-AD to test these mixtures, determining if a specific population can be modeled as a combination of others. When QP-AD is applied to the Swat Valley timeline from 1200 BCE onward, the statistical results become definitive. The model confirms that the earliest Swat samples contain a roughly 22% introduction of Middle to Late Bronze Age steppe ancestry. This ancestry carries the R1a Z93 haplogroup, a specific male lineage marker associated with the spread of Indo-European languages. This entire genetic package, both the autosomal DNA and the male haplogroup, is absent from the preceding Harappan phase. This integration matches the archaeological timeline of southward expansions from the steppes between 2000 and 1500 BCE. Advanced distal modeling confirms the arrival of steppe ancestry at the subcontinent's threshold, aligned with the traditional timeline for the Indo-Aryan migrations. The archaeogenetic record shows that before 1900 BCE, the specific genetic signature of the steppe does not exist in South Asia. Around 1900 BCE, a long-term drought led to the drying up of river systems and the systemic collapse of the Indus Valley urban centers. The abandonment of these cities triggered a demographic restructuring of the entire subcontinent. One major migration moved south and east, away from the original Harappan core. These southward moving groups mixed with indigenous AASI populations who had not encountered West Eurasian gene flow. This mixture formed the Ancestral South Indian baseline, a population that initially carried no steppe ancestry. Simultaneously, Harappan descendants remaining in the northwest integrated with the arriving steppe pastoralists. This systemic collapse fractured the Harappan population into two new foundational mixing pools. The second pool formed the Ancestral North Indian baseline. This group integrated the northwestern Harappan heritage with the signature of the western steppe herders. These Bronze Age mixing events created the genomic structure of the 1.5 billion people living in South Asia today. This relationship is mapped on a principal component analysis plot. The Ancestral South Indian and North Indian baselines act as two anchoring poles on opposite sides. When modern South Asian genomes are plotted, they align along a strict linear continuum between these poles. Every individual on this gradient is a mixture of these original pools, confirming the dual mixture model. Analyzing these distal components reveals the specific moments these lineages converged. The woman from Rakhigarhi anchors the Harappan origin and traces their genetic legacy into the present, a bedrock population integrated with Bronze Age steppe DNA.